We're back with review session 3, and starting on page 678, figure 1730, and looking then at the regulation of bile release. You guys know the drill now, so go ahead, take a look on that figure. Notice, first of all, we have chyme containing fat coming into the duodenum from the stomach. Number 2, then, cholecystokinin is being released. And this, if you recall, is the third time that we're seeing CCK. So we've already talked about the fact that CCK inhibits gastric motility. We've talked about the fact that CCK stimulates the release of pancreatic juice rich in digestive enzymes. And now notice, if you follow the arrow from the bottom all the way up to the upper left, now we're talking about the fact that CCK also has an effect on the gallbladder. And when CCK reaches the gallbladder contracts, squeezes its stored bile out. And so remember, basically all that the gallbladder is doing is storing and releasing bile, well, and concentrating it. But basically, the gallbladder doesn't make the bile. Remember, this is made in the liver, but the gallbladder largely functions in storing bile. And so now, as a result of CCK getting to the gallbladder, the muscular wall of the gallbladder constricts, the gallbladder squeezes bile out into the cystic duct. Again, cystic duct, remember, coming away from the gallbladder, and you'll recall then that the pathway is on down to the duodenum. And now we have bile entering the duodenum and able to do its job with respect to fat molecules. So, functions of bile salts, back to your lecture outline, and bile salts then aid digestive enzymes by breaking up fat clumps into smaller fat droplets, the process called emulsification. And again, you'll recall we initially talked about emulsification at the beginning of this chapter. Emulsification then basically not digesting fats chemically, but instead just increasing the total surface area of the fats, taking a big clump of fat molecules, if you will, and breaking it down into smaller clumps. And this then gives lipase molecules better access to the fat molecules, increases the surface area, and digestion of fats can then occur more quickly as results. And then notice again, back to your lecture outline, bile salts also enhance the absorption of fatty acids and of cholesterol and of fat-soluble vitamins. And so all these things we're talking about that bile does relate to lipids. From here, we're getting into 17.8 now in your textbook and looking at the structure of the small intestinal wall. And remember then, we talked about the fact that the wall of the small intestine, the inner lining actually of the small intestine, has several series of progressively smaller folds, and each of these three series of folds increases surface area. And the idea is that with this vastly increased surface area of the small intestine, absorption of nutrients can occur more quickly. We looked then, first of all, on page 682 at figure 1738, we looked at these large folds, these big folds, of the lining of the small intestine. And remember, we called these folds plicae circulares. We then looked at medium-sized folds, if you will, along the surface of the big folds, along the surface of the plicae, and these medium-sized folds then are the villi. And then finally, flipping back a page to page 681 and looking at the diagram of a single cell, upper right-hand part of the page now, looking at the diagram of a single cell that's part of the lining of a villus. Notice we have these small folds along the medium folds. Again, the villi are the medium folds, and these small folds now along the surface of the individual epithelial cells. These small folds are called microvilli. Going on to talk now about secretions of the small intestine, and let's just run through this portion of your lecture outline. So we have goblet cells and we have mucus secreting glands, or Brunner's glands. Both of these, of course, goblet cells and mucus secreting glands, obviously secreting mucus. Goblet cells in the mucosa, you'll recall from lab. Brunner's glands, we didn't get to the point in lab where we got to see these, but Brunner's glands in the submucosa, and goblet cells and mucus secreting glands, both secreting mucus into the lumen of the small intestine. Again, largely just to lubricate and to help digesting food move along. We also have these glands called intestinal glands, or crypts of Lieberkühn, at the bases of the villi, and you may recall these from lab, and again, these were in that enlarged area of the model of the wall of the small intestine. And these intestinal glands then secrete a watery fluid. And I did notice here on your lecture outline, the next sentence is not worded correctly. So if you're to that point where it says, after it says secrete a watery fluid, where it begins the next sentence with enzymes embedded in, you can cross those three words out. So cross out enzymes embedded in, and then just capitalize the T in the, and then we're good to go. So we're saying now the epithelial lining of the mucosa secretes, add an S here, so the epithelial lining of the mucosa secretes digestive enzymes, including peptidases, which split peptides into their component amino acids. So take these fairly small chains of amino acids and break them down into single amino acids. Also secreted by the small intestine, lipase, again, breaking down triglycerides, breaking down fat molecules into fatty acids and glycerol. And then also the small intestine is secreting this series, this group of enzymes called sucrase. The enzyme is sucrase, and another enzyme called maltase, and a third enzyme called lactase. And these respectively then break down, and so sucrase breaks down sucrose, maltase breaks down maltose, lactase breaks down lactose, and you'll recall sucrose, maltose, lactose are all disaccharides, and these enzymes break them down into their component monosaccharides. Back to your handout then, the small intestine also produces enterokinase, converting trypsinogen into trypsin. Go back to the pancreas for trypsin. And then the nutrient molecules mix with fluid from the intestinal glands, and then this fluid from the intestinal glands, now containing these mixed small nutrient molecules, this liquid, this fluid is now reabsorbed. And so now we've accomplished the objective of all of this, which remember is to get small nutrients out of the alimentary canal and largely to get them absorbed out of the small intestine and to make them available via the bloodstream to cells throughout the body. In terms of regulation of small intestinal secretion, recall, and this should be in your notes now, two things stimulate secretion by the small intestine. One is contact with chyme. So chyme comes into the small intestine. This stimulates the secretions of the small intestine, the release of those secretions. And number two then, parasympathetic impulses, again parasympathetic, rest and digest, resulting from the distension, the expanding of the intestinal wall. So the intestinal wall simply stretches a bit as food enters, and this stimulates those nerve impulses. In terms then of movements of the small intestine, we have mixing movements, and again you can refer back to page 657 for a diagram of these mixing movements within the small intestine specifically. And then we have propelling movements, and we do have peristalsis going on within the small intestine. And the peristalsis then, as you should have in your notes now, involves short, weak waves of peristalsis, resulting in kind of a continuous, slow movement of digesting food through the small intestine. We'll end here and pick up in review session 4 with the large intestine.